All right, I want to welcome you out to chemistry. This is going to be our first unit of the year, known as measurements and uncertainties. We're going to start off with our first lecture, Lecture 1, under measurements. And you can follow along this lecture with your lecture notes that are passed out in class. Make sure you take really good notes and study those notes to prepare for the quiz for your next class. All right, we're going to start off with the essential question of this lecture. What are the major sources of error in experimental measurements? You need to be able to answer this question by the time we are done with this session here. All right, let's go on. Right. What exactly is a measurement? Let's write down kind of a definition. A measurement is more or less a quantitative observation that always consists of three parts. The three parts is a number, must always have a number, a uncertainty, and a unit. If you do not have these three parts in your measurements that you make in the lab or on a test or homework or whatever, it would be considered wrong. So you always must, again, contain a number, an uncertainty, and a unit in order for it to be a correct measurement. All right, so in 1960, an international agreement was reached specifying a particular choice of metric units for use in scientific measurements. These units are called SI units. The SI units are listed in page 7 in this handout that you have, and it should be labeled under SI units. Become familiar with those SI units, and specifically come familiar with the SI prefixes, which are listed on page 7. All right, now we're going to get into something that's really important and sometimes is hard or difficult for students to first understand. The idea between accuracy and precision. All right. Often we get those two words usages mixed up. Sometimes we refer accuracy when it's really supposed to be precision, or something being precise, or we use precision when we really mean accuracy. And so let's make sure that we understand these, because we're going to be discussing accuracy and precision throughout the year. Okay? So the two important concepts to understand are the differences between accuracy and precision. So first of all, accuracy refers to how closely individual measurements agree with a generally correct or accepted literature value, something that the scientific community feels is correct or true. And that is what accuracy deals with. So for instance, if the boiling point of water is 100 degrees Celsius, and you measure the boiling point of water in an experiment with, say, a digital thermometer, and it reads 99.98 degrees Celsius, it's really close to the true value, so you would say that your measurement is accurate or very close to the true value. Right? However, precision indicates how many significant figures there are in a measurement. Okay? Now in our next tutorial we're going to talk more about what significant figures are, but every instrument, every measuring instrument that you use in the lab to measure a measurement or something is going to have a limitation. And the limitation will only allow us to be able to record certain number of digits for that measurement. And the more digits, or in other words, the more significant figures that can be recorded for a measurement, it means our measurement becomes more precise. And so in class we'll look a little bit more about what that entirely means. I got an example here for you. All right, here's our example. The normal boiling point of water, again, is 100 degrees C. Oops, go back one. Okay, normal boiling point for water, again, is 100 degrees C. So if a mercury thermometer could measure the normal boiling point of water, and it measured to 99.5 degrees C, whereas you used a data probe to record the temperature of water, and it was 98.15 degrees C. Well, if you look at these two measurements, you'll find that the mercury thermometer measured our temperature of water more accurate. It was closer to the true value of 100 degrees C. So our mercury thermometer is more accurate. However, which one is more precise? Well, which one of these measurement devices, the data probe or the mercury thermometer, allows us to have more significant digits? Well, it turns out it's the data probe. The data probe lets us have 4 significant digits, as you can see, the 9, the 8, the 1, and the 5, whereas the temperature or the mercury thermometer only allowed us to record 3 significant digits, the 9, 9, and 5. So therefore, again, we would say that the mercury thermometer is more accurate because it gives us closer to the true value of the boiling point of water at 100 degrees C. But the data probe is more precise because we can measure more digits or significant figures. All right, next what we need to do is, when it comes to accuracy, there is a calculated value in which we can determine how far we are away from the true value when we do an experiment. So let's write down this expression or equation in our notes here. So first of all, to find the percent error. Percent error. And the percent error, again, is just going to tell us how far away our experimental value is from the true literature value that is accepted by the scientific community. And you're going to have to learn how to calculate that when you do labs. So we're going to take our experimental value, you're going to take our experimental value and you're going to subtract that from the accepted values. Sorry, it's going out of the box there. Okay, So again, we take our experimental value, the value that we calculate in the lab, and we subtract it from the accepted value, and we're going to divide that by the accepted value. And then to get into a percentage, we're going to multiply this whole thing by 100. So make sure you record this equation in your notes and memorize it. And we're going to give you some problems to solve in class using this equation. And of course, when we do labs, you will also be expected to use this equation to find your percent error, or again, how close your experimental value is to the accepted value. As it says, you should compare your results to literature values when appropriate using this expression above. The accuracy of a measurement depends on the experimental design or procedure on the carefulness of the measurements. All right, and we'll spend some more time with that in class. All right, so we're going to go on to our next part, and that's on experimental errors. In chemistry, it's really important that when you do laboratory work and you analyze your data and the results that you get from a lab, that you're able to identify all of the errors that occur in your lab. Now, experimental errors are always going to occur, always. And so we're going to talk about what type of experimental errors there are and what are some possible reasons for those errors. All right? All right, so first of all, the first experimental error we're going to look at is known as random error. Let's go back a slide here. There we go. So the first is random error. And random error arises from limitations in the instrument's precision. And can lead to readings being above or below the true value. Now remember that measuring instruments can only record to a certain number of significant figures because of its limitations. And so instruments can only have certain precision, and precision problems lead to random error. Therefore, random error will always occur in any measurement. We can never, ever eliminate random error. And that's important to remember. Again, random error will always occur. Can't eliminate it. This is because the last digit of any measurement is always estimated. Okay? Always estimated, the last digit. And you will learn more about that when we talk specifically about significant figures in measurements in our next tutorial. You can decrease random error, though, by making careful measurements, by using a more precise instrument, and that means a more precise instrument has more significant digits or figures that can be recorded, and repeating an experiment a number of times. Usually our experiments we're going to repeat 3 to 5 times so that we can reduce our random error problem. So I really want you to pay very close attention to these notes here. Random error is really important for you to understand. So if you need to go back and look at this tutorial, do so. Study these notes on random error, and be ready for your quiz on that. All right. All right. So here's an example we got here. We're going to look at precision and random error a little bit. So we have a ruler, and you will notice that the ruler is going to have some limitations. There are certain values that we're all going to agree on when we use this ruler and measure this piece of wood. But then there's going to be, which is the last digit, is going to be estimated, which we won't agree on. Okay, I think everyone will definitely agree that the first digit is, the first digit is going to be 4. Okay, so we know that the piece of wood is greater than 4, but less than 5. And then we can go ahead and get the next digit. We can count 1, hash mark, 2, 3, 4, 5. And it looks like it's going to be in between 5 and 6. So we can go 4.5. Because the next uncertain digit is between 5 and 6, we're going to have to estimate that digit. Now it is important, we do have to include one estimated digit in every measurement. So we're going to estimate that I am probably about a 7. You might estimate it as a 5 or a 6 or some other digit, and that's okay. And so according to this ruler, we can only measure 3 significant digits. So our instrument on this, or the precision on our instrument ruler, is only 3 significant digits. That's it. We cannot add another one. In any measurement, we can only have one estimated digit, and that in this case is my 7. Okay. Now because of that, I have random error. I do not know exactly for sure what this digit is. It's completely being estimated. And that will introduce my random error. Alright, this next slide we're going to spend a little bit more time talking about in class, and talking about how we address.
Good morning, Hillcrest. This is Caleb.
Okay, so go ahead and read through this slide. Learn it as much as you can, and if you have questions, again, we'll talk about this more in class on this slide. And then it is an important slide. Alright, so the next type of error is going to be systematic error. Okay, systematic error occurs in the same direction each time. It is either always high or always low, and always deviating from what we say the accepted or true value. So systematic error, these are errors that's going to occur, that's going to cause us to get away or deviate from our true value or the accuracy of the experiment. A lot of these systematic errors results in flaws or defects in instruments. That would be your measuring instruments that you would use during the lab, or from poor experimental design or the procedure. This is something we're going to look heavy at when we do our labs is our experimental design, our experimental setup, and where errors could come into place in our design, what might deviate us from the true or the accepted value. Alright, so that's the end of our tutorial for today. Make sure you go back and relook at this if you need to. You can play it over and over again. Make sure you have good notes written down. Study those notes and be ready for a quiz in class.